Hello everyone and welcome to our module on neoplasia. Neoplasia literally means new growth, and this is the pathology term for cancer or malignancy. And you should be aware that all cancers are ultimately genetic disorders. They all start at the gene level with a gene abnormality, and that's what leads to cancer. And the idea here is that the cell cycle, which I talk about in detail in the cell biology section, is normally under tight control. Certain signals lead to cell growth and cell division. Certain other signals lead to the prevention of excessive growth or in cell division. So if there's a mutation in one of the genes that helps to control the cell cycle, that leads to uncontrolled growth, and that leads to cancer. One of the underlying principles of neoplasia or cancer is clonality. So the idea here is that a single cell develops a genetic mutation that allows it to grow uncontrollably. This is what leads to a tumor, because now that single cell gives rise to daughter cells, which are called clones, and all of those clones carry the same mutation. So this is shown in this picture on the screen. You've got a cell that's dividing normally, and then one becomes damaged and develops some abnormal DNA. That should lead to apoptosis, and in many cases it does, but if the cell does not die, it then begins to grow uncontrollably without the ability to stop like normal cells, and that's what leads to cancer. The genetic mutations that are found in tumor cells can change over time as the cells go through more and more levels of division. This is a principle called tumor progression. So let's imagine that we have a cell here in red and it's got a gene mutation and it's dividing uncontrollably. During one cell division, another mutation may occur because now this is a rapidly dividing cell, dividing in an uncontrolled manner, and maybe another division leads to a mutation that causes the blue cell up here. What this means is over time, the cells are going to accumulate a multitude of different mutations, and this is going to make each cancer cell slightly different from the other. And this is a problem because it sometimes makes cancers less responsive to chemotherapy. Maybe, for example, the chemotherapy works on the red cells here, but not the blue ones. That's going to be a problem. And so the point here is that large tumors, which have cells that grew out of multiple cell divisions, are often heterogeneous in terms of the mutations that you find in each cancer cell. Once you appreciate that malignancies are the result of disorganized cell growth, then it's easy to understand the common tumor locations. There are many places in our body that even under normal conditions contain rapidly dividing cells, for example, the epithelial layer of our GI tract, glandular tissue in the breast, or skin cells. In all of these places, there are cells that even under normal conditions are stopping and starting cell division regularly. This means those cells are constantly copying their DNA to make daughter cells. Each time they copy their DNA, there's a chance for a mutation to occur, and therefore these cells have a higher likelihood of developing into cancer. Once you get this, it's easy to appreciate that the GI epithelium is a common site of cancer. On the other hand, the myocardium, where cells do not normally divide, is a very rare site of cancer. This all has to do with how much cell growth occurs under normal conditions in these locations. Cancer cells metabolize glucose in an unusual way. This is known as the Warburg effect after the person who first described it. So in cancer cells, glucose is metabolized to lactate to generate ATP, even if there is oxygen around. This is very unusual. It's called aerobic glycolysis because the cells are using only glycolysis even though there's oxygen around, and they could use oxidative phosphorylation. In the biochemistry section in the video on glycolysis, I talk about how glycolysis metabolizes glucose to pyruvate, which can then enter the TCA cycle to generate ATP through oxidative phosphorylation. Cancer cells don't do this. Instead, the pyruvate is metabolized to lactate even under aerobic conditions. This does generate ATP for cancer cells, but it's less than oxidative phosphorylation. And as I said before, this occurs even when oxygen is present. The reason why cancer cells do this is not understood. But the result is that cancer cells have to take up lots and lots of glucose to generate enough ATP because they are less efficient at generating ATP per glucose molecule compared to if they use the TCA cycle and oxidative phosphorylation. And the fact that cancer cells take up lots and lots of glucose is useful clinically because it's the basis of something known as a PET scan. This is a scan that uses radio-labeled glucose in order to detect cancer and cancer mets. This is a picture of a PET scan. This person has a malignancy in their abdomen. You can see how brightly it's lighting up. The reason it's so bright is because it is taking up lots and lots of radio-labeled glucose, and it's because of the Warburg effect and the unusual metabolism in cancer cells that we can do a PET scan. It's often used to identify tumor mets because distant metastases will light up on a PET scan. In the video on cellular adaptations, I talk about dysplasia. This is a description of tissue morphology. Dysplasia means disordered but non-neoplastic growth, and this is a change in tissue beds that often precedes neoplasia. In other words, the tissue begins to display dysplasia, and if the dysplasia continues, eventually cancer will develop. In other words, it progresses to cancer. This is usually described in epithelial tissues like the GI epithelial layer or breast tissue, and sometimes the term carcinoma in situ is used. And what this term means is that a tissue bed is displaying dysplasia of the entire epithelial layer with no invasion of the basement membrane yet. In other words, the carcinoma or the dysplasia is contained within the epithelial layer. It has not broken through the basement membrane. So here's a cartoon to show you how cancers often develop. So you've got some rapidly dividing cell layer. Maybe it's the GI epithelium, maybe it's your skin cells, and then one of the cells develops a mutation that allows excessive growth. This will often first lead to hyperplasia, just excessive growth. Eventually, the tissue bed will become dysplastic, meaning it will display abnormal morphology under the microscope. That will progress to in situ cancer, which means dysplasia and maybe malignancy cells that do not invade the basement membrane. And then eventually, there will be an invasive cancer that breaks through the basement membrane. And this is the typical progression from a gene mutation all the way to invasive cancer. Next, let's talk about anaplasia. Anaplasia is a very, very bad thing. You never want to have anaplasia of any of the tissues in your body. Anaplasia means undifferentiated cell growth. It means that the cells under the microscope do not look at all like their cells of origin. So imagine a normal cell, for example, an epithelial cell in the GI tract. It has certain features that give it a certain shape, and it has certain cell proteins and cell surface markers. What happens in anaplasia is a GI epithelial cell, for example, becomes so malignant that it directs all its metabolic activity to growth. It's doing little or no other functions. What this means is when you look at it under the microscope, it won't look at all like an epithelial cell. For that reason, anaplasia is seen in malignant aggressive tumors. It usually indicates a very poor prognosis. And the idea here is one of the basic principles of cancer pathology. Well-differentiated tumors look a lot like their tissue of origin, and this is a very good thing. It means that the cell is not devoting all its efforts to growth and malignant spread. It's still devoting some of its efforts to looking like the tissue of origin and performing the normal functions of the tissue of origin. In contrast, anaplastic tumors have a lack of distinguishing features, and this is very bad because all they're doing is growing. There are a number of changes in cellular physiology that must occur for a cell to become malignant. These are referred to as the hallmarks of malignant cells, and I've listed some of them on this slide. The first one is autonomous growth. So malignant cells are not sensitive to growth factors and inhibitors. As we've been discussing, they are going through the cell cycle in an unregulated manner, and therefore they don't respond to the normal signals that control growth. The second hallmark is evasion of cell death. So cells with abnormal DNA often die through a couple of mechanisms. They can either undergo apoptosis, or sometimes they're killed by cells of the immune system. For reasons that are poorly understood, this doesn't happen to malignant cells. They do not undergo apoptosis even though they have genetic mutations, and they are able to evade the immune system. Malignant cells also must have unlimited ability to replicate. This is referred to as being immortal. Normal cells become what's known as senescent after a certain number of replications. We'll talk about why this happens in a minute, but the point is that they cannot divide indefinitely. After 50 or 60 divisions, they become senescent and are unable to divide any further. Malignant cells, on the other hand, are immortal and they have the ability to divide indefinitely. That's why tumors can grow and spread. The next hallmark is angiogenesis. If tumors could not develop new blood vessels, they wouldn't be able to grow. They would outstrip their blood supply. But all malignant cells have the ability to stimulate new blood vessel growth, and this is what allows them to get fuel to grow. And then finally, they must have the ability to invade tissues and spread. This is what makes malignancies malignancies. In some of the DNA videos in the cell biology section, I talk about the enzyme telomerase, and this is important for understanding cancers. Normal cells are capable of only about 60 to 70 divisions, and thereafter they become senescent. And the reason they can only divide this number of times is because each time they divide, their telomeres shorten a little bit. The telomeres are nucleotides at the end of chromosomes, and they get shorter with each cell division, and eventually they become so short that the cell stops dividing. The enzyme telomerase avoids the loss of genetic material with duplication, and this stabilizes the telomeres. And this enzyme is active in stem cells so that they can continually divide to replace other cells. In other cells that are not stem cells, there's very little activity of the enzyme telomerase. It turns out that in almost all cancers, this enzyme is upregulated for reasons that are poorly understood. But for the point of your board exams, you should understand that cancer cells need telomerase if they want to keep dividing indefinitely and not become senescent after a certain number of cell divisions. Now let's talk about the formal system clinicians use for describing a tumor. All tumors have what's known as a grade. This represents the degree of differentiation. This is always determined by a pathologist, and it requires a biopsy for microscopic tissue analysis. And it's high yield to remember that this requires a biopsy. A basic step one question you could see is, how is a tumor grade assigned? And you need to know it's not done from a CAT scan, it's not done from a physical exam or a blood test, it's done from a biopsy examined by a pathologist. Pathologists will give tumors a grade of 1, 2, 3, or 4. A very well-differentiated tumor will receive a low grade, and that's generally good. An anaplastic or undifferentiated tumor will receive a high grade, and that's often bad. Another way we describe tumors is by stage. Stage refers to the degree of tumor extension or spread. For example, whether the tumor is localized to the breast or has spread to lymph nodes or has distant metastases. These things determine the stage. And in contrast to grading, stage is usually determined by radiology or imaging. For example, many patients with cancer will undergo a head-to-toe CAT scan looking for evidence of spread of the tumor. When we say a tumor is early staged, we mean that it has localized growth confined to the tissue of origin, for example, the breast. When we say a tumor has an advanced stage, we mean it has spread and often has distant metastases. And in many cases, stage is the most important prognostic factor. The reason is that early stage cancers can often be completely removed surgically. On the other hand, tumors that have metastasized often can't be treated surgically and must be treated with chemotherapy. On your clinical rotations, you'll often hear clinicians use what's known as the TNM staging system, so let's talk about that now. The TNM staging system gives each tumor 3 numbers, a T number, an N number, and an M number, and each one means a certain thing. So the T number is for the primary size of the tumor. T1 tumors are relatively small and T4 tumors are relatively large. N is for the degree of regional lymph node spread, and 0 means no lymph node spread, and 3 means extensive lymph node spread. And then finally, M is for metastases. M0 means no distant metastases, and M1 means there are distant metastases. So for example, a tumor could be T4, N2, M0, and using this system, you would know what that means. Tumors can be classified as benign or malignant. Benign tumors are likely to remain localized without spread. They're also usually amenable to surgical removal. Now, this doesn't mean they won't cause problems. A tumor that is not spreading but is large and is compressing a structure like the trachea can be very dangerous. Benign just means it's unlikely to spread. It doesn't mean it can't cause other problems. Benign tumors are also usually well differentiated, and when you look at them under the microscope, they usually display low mitotic activity, meaning they are not dividing very rapidly. In contrast, malignant tumors are basically the opposite. These are tumors that tend to invade and spread. They may cause death. They're often difficult to treat solely with surgical removal. They often require chemotherapy and radiation therapy. Every once in a while, you will find a patient who has a mass. Maybe it's a mass in their abdomen, maybe it's in their lungs. And when the mass is biopsied, it turns out that it is an abnormal growth of cells, but it is not neoplastic. There are basically 2 times when this happens. The first time is when someone has a hamartoma. This is a mass of mature but disorganized cell. For example, patients often develop lung hamartomas. These growths contain lung tissue, but it's disorganized and different from the rest of the lung. This is basically a developmental anomaly. The lungs grow entirely normally, but in one place they abnormally grow, and this leads to a clump of normal, healthy lung tissue. It's not malignant, it's not neoplastic, but it is still a mass and it is still abnormal. Second example of a non-neoplastic growth is a choristoma. This is a mature, well-differentiated tissue, but it's in the wrong place. So there's nothing neoplastic about the tissue. It's not going to grow and spread, but it's in the wrong place. And the classic example of this is a Meckel's diverticulum, which I talk about in the GI embryology videos. Meckel's diverticulum is gastric tissue, but it's found in the ileum, which is part of the small bowel. Once again, nothing neoplastic, just mature tissue in the wrong place. And both of these are benign, meaning they do not invade or metastasize. Let's talk about the way that tumors are named, and we'll start by talking about benign tumors. Benign and malignant tumors are named differently. In terms of benign tumors, they are often named by the cell or tissue type of origin plus the word oma. So for example, a fibroma is a benign tumor that derives from fibrous tissue. A chondroma is a benign tumor that derives from cartilage tissue. In many organs in the body, adenomas form. These are benign tumors that derive from epithelial cells, and the term adenoma is usually applied when it involves glandular structures. Either the tumor derives from gland cells or the tumor resembles gland cells. So for example, you can see adenomas in the adrenal glands and in many other locations in the body. And then finally, papilloma is another term used for benign tumors. These are also epithelial tumors, but they get the name papilloma because their surfaces have finger-like projections, and that's what a papilla is. It's a finger-like projection. Malignant tumors are named a little bit differently. If the tumor derives from mesenchyme, that means if it derives from connective tissue, bones, blood, or lymph, it's named with a certain system. Solid tumors which derive from these locations are called sarcomas. So for example, an osteosarcoma is a malignant mesenchyme tissue that derives from bone. That's why it has the word osteo in it. On the other hand, if the mesenchyme tissue is blood or lymph, it's called a leukemia or a lymphoma. And then just like with benign tumors, epithelial tumors, because they're so common, have their own naming system. So malignant epithelial tumors are always called carcinomas. If they derive from glandular tissue, they're called adenocarcinoma. So in other videos, I talk about colon adenocarcinoma or lung adenocarcinoma. These are carcinomas, which means malignant tumors of epithelial cell origin deriving from glandular cells. On the other hand, in the skin, you can find a squamous cell carcinoma. This means it's a malignant tumor. It's a carcinoma. It's a malignant tumor of epithelial cells. In this case, it comes from squamous cells. All tumors can spread locally by just growing larger and invading the tissue that surrounds them. But in order for a cancer cell to become a distant metastasis from a tumor of origin, it has to travel either through the bloodstream or the lymphatics. Sarcomas always spread through the blood. In other words, they always have hematogenous spread. And in addition, they always travel through the veins. The reason for this is because arteries have thick walls that are difficult to penetrate, but the veins have thin walls and therefore they are easily penetrated. And because these tumors spread through the venous system, they often end up in the liver and the lungs. All of our venous blood flow goes into the lungs and much of it goes into the liver, so these are common locations for hematogenous spread and metastases. Carcinomas, on the other hand, usually spread via lymphatics, except for 4 classic exceptions. And as a med student, you should definitely know these 4 exceptions. It could come up on your boards, and it's always asked on clinical rotations. So choriocarcinoma, which is a placental tumor, displays early hematogenous spread. It's a carcinoma, but it travels through the bloodstream. Renal cell carcinoma is also famous for invading the renal vein and traveling through the bloodstream. Hepatocellular carcinoma commonly invades the portal vein and therefore travels via the bloodstream. And then finally, follicular thyroid carcinoma is another carcinoma that travels via the bloodstream. Let's talk about teratomas. These are a special form of malignancy that contain more than one cell type. So they don't just contain skin cells or lung cells or muscle cells. Instead, they contain multiple different cell types. And importantly, the different cell types come from multiple germ layers. So for example, there may be some cells like skin or hair follicles, which are from the ectoderm germ layer. Other cells may be from the lung or the GI tract, which is part of the endoderm, and still more cells may be muscle or cartilage, part of the mesoderm. Importantly, all the germ layers are represented within the different cell types of a teratoma. And the reason they have so many different cell types is because they arise from germ cells in the ovaries and testes, and these cells are capable of developing into multiple different cell types, and that's why they form all the different cell types of the teratoma. Let's talk about cancer epidemiology. For adults in the United States, cancer is the second leading cause of death. Heart disease is number 1, respiratory disease is number 3, diseases like COPD in particular, and accidents or trauma are number 4. In terms of cancer incidence, meaning new cases of cancer each year, the number 1 new type of cancer is breast cancer for women or prostate cancer for men. The second most common new type of cancer is lung cancer for both genders, and colorectal is third for both genders. The mortality rate from cancer, however, the death rate is different. In terms of the death rate, lung cancer is the most deadly of all the cancer types for both men and women. Second most deadly is breast cancer for women and prostate cancer for men, and the third most deadly is colorectal cancer. The epidemiology of cancer is different in children compared to adults. First of all, the causes of death are different for children. The most common cause of death among children is accidents, and if you've ever watched little kids play, you would know why this is the case. Second most common cause of death is cancer, and third is congenital disorders. In terms of both the incidence and the mortality from cancer, leukemia is number 1. In particular, acute lymphoblastic leukemia is very common in children. Second cancer in terms of incidence and mortality are CNS tumors, and third is neuroblastomas. I'll finish this video by talking about carcinogenesis, which is the process by which cancer begins. As I told you before, cancer is ultimately a genetic disorder, and what this means is in order for a normal cell to become a malignant cell, it must acquire some type of non-lethal DNA damage. The DNA damage could be an error in copying DNA. It also could be an error caused by something outside the cell like heat or radiation. We'll talk more about these things. They're called carcinogens later in this video. But importantly, the damage must be non-lethal. As we talked about before, cancer cells are able to avoid death by apoptosis or by the immune system. So if a normal cell acquires some DNA damage that is lethal, then it won't become cancer. It must acquire non-lethal damage, and that can lead to cancer. And although there are many different genes that can lead to cancer, they are generally classified as one of 2 types. Genes that cause cancer are either tumor suppressor genes or oncogenes. A mutation in one of these two classes of genes is what typically leads to cancer. Tumor suppressor genes are genes that limit cell growth. A classic example is the p53 gene, which I talk about in the video on cell cycle in the cell biology section. This gene codes for a protein that blocks progression through the cell cycle, so it normally stops cells from dividing. If it is mutated, the cell will divide uncontrollably, and that's why it's called a tumor suppressor gene, because its normal role is to suppress cell growth. Another tumor suppressor gene is the retinoblastoma gene. This becomes mutated in the form of eye cancer called retinoblastoma, and the protein from this gene inhibits transcription factors. So if this gene becomes mutated, those transcription factors become uninhibited, and the cell can begin to grow uncontrollably. Now, importantly, and it's very high yield to understand this, for all genes in our body, you have two alleles. Let's call one big A and the other little a. I talk about this in detail in the genetics videos. In order for a mutation in a tumor suppressor gene to convert a normal cell to a malignant cell, there must be mutations in both alleles. If one allele, for example the big A, were mutated, the other allele could continue to produce the proteins of the tumor suppressor gene and the cell would not grow uncontrollably. There must be a mutation in both genes in order for the cell to become malignant. Germline mutations are mutations in genes that are found in all cells in the body of a baby at the time of birth. And the reason they're called germline is because they are a mutation in a gene that occurs very early on in development of the embryo. So that gene becomes mutated and it begins to produce offspring, and all the offspring in the embryo and fetus will carry that gene mutation. Sometimes one of these mutations occurs in a tumor suppressor gene, and when this happens, it makes it very easy for cancer to develop, because all the cells in the body already have one allele that is mutated, thus they only need to acquire a second mutation, and that will lead to malignancy. And this is the case in a number of genes involved in hereditary forms of cancer. I talk about these in lots of other videos. The BRCA1, BRCA2 genes in breast cancer can be inherited as a germline mutation. Same is true for hereditary forms of retinoblastoma. HNPCC or Lynch syndrome and FAP syndrome are both colon cancer syndromes. And finally, Li-Fraumeni syndrome is a syndrome involved with multiple malignancies. But all of these involve a germline mutation, which makes it very easy for a cell to acquire a second mutation of a tumor suppressor gene, because all cells are born basically with one mutated gene. In contrast to tumor suppressor genes, oncogenes are genes that promote uncontrolled cell growth. They do not suppress it. And many of the genes in the cells in our body are what's known as proto-oncogenes. This means they have a normal role, but if they become mutated, they can lead to cancer. So some examples of proto-oncogenes are genes that encode for growth factor receptors, or signal transducers. These all have a normal function in the cells of our body, but if they become mutated and overactive, they now stop being a proto-oncogene and they become an oncogene, and that's what leads to cancer. And a high-yield thing to understand about oncogenes is you only need a single gene mutation to lead to malignancy. Once again, let's imagine we have two alleles of an oncogene, big A and little a, and let's imagine that these alleles code for a growth factor. If one of them becomes mutated, let's say it's this one, it can begin overexpressing that growth factor, and that alone will lead to cancer. The fact that the other allele is normal will not protect the cell the way it does with tumor suppressor genes. I'll finish this video with this slide and briefly mention carcinogens, which are substances that can cause cancer. And in other videos on specific cancer, I talk about these. But in general, these are things that are often outside the cell that can cause a cell to become malignant. So for example, some chemicals are carcinogens. Asbestos leads to the lung tumor mesothelioma. Many viruses are carcinogenic. For example, HPV can lead to cervical cancer. Epstein-Barr virus is also carcinogenic in many cases. And as you likely know, sunlight can cause cancer, and that's because sunlight contains radiation, another classic carcinogen. And that concludes our video on neoplasia.